Hi, everyone. My name is Abigail Newbury, and I'll be presenting our work in NPJ Digital Medicine titled Multi-Domain Role-Based Phenotyping Algorithms Enable Improved GWAS Signal. This is work between myself, my collaborator Ahmed El-Hussein, and our PI Gamze Gursoy, and we are co-appointed with the New York Genome Center and the Columbia University Department of Biomedical Informatics. So I want to start off by talking about genome-wide association studies, which link genomics and healthcare data. And so in a very simple GWAS, we're regressing a phenotype vector Y on our genetic data matrix G and some covariate matrix Z. And so our genotype matrix is typically made of samples by SNPs, and SNP here is a single nucleotide polymorphism. And so this represents a position along the DNA in which someone can have 0, 1, or 2 copies of an alternate allele. And for covariates Z, we're typically including things like age, sex, and principal components of G, which are shown to represent axes of genetic ancestry and can help to adjust out confounding, because this is a very common confounder in genotype-phenotype relationships. And so once we perform our GWAS, insights that we can glean from this are that we can identify associated SNPs. These are SNPs significantly associated with disease that might help us understand a bit more about biological mechanisms of disease. And we can also aggregate these SNPs and their effect sizes in order to attempt to predict individual disease risk based on genetic information with things like polygenic risk scores. And so we know that phenotype misclassification can affect GWAS results. So in a very simple regression of a binary phenotype on a binary risk factor, we'll observe an effect size that's diluted from the true effect size proportional to the positive and negative predictive value of a phenotyping algorithm. And so in simulations, it's been shown that when we increase false negatives, our effect sizes bias towards the null, and when we increase false positives, our effect sizes can bias either towards or away from the null. And so here we obviously know that accurate EHR phenotyping will reduce type 1 and type 2 errors in GWAS. And so with all of that, the goal of this work was to assess the impact of various role-based phenotyping algorithms on GWAS outcomes, examining factors such as power, heritability, replicability, functional annotations, and polygenic risk score prediction accuracy. And we performed this work in the UK Biobank across 7 diseases. These are Alzheimer's, asthma, COPD, myocardial infarction, rheumatoid arthritis, systemic lupus, and type 2 diabetes. And we performed this work across 4 different open source, commonly used role-based phenotyping definitions. So to get a bit more into the algorithms used, we use rule-based phenotyping algorithms with varying levels of complexity. So the first I'll talk about we refer to as ADO. This is UK Biobank's algorithmically defined outcomes, and these are defined for 15 diseases, and they're really highly curated. So a group of clinical experts for each disease creates these algorithms, and they include 1 or more occurrences of conditions in the condition occurrence table, but also in the death table. And then for 3 of the 4 algorithms also include occurrences of self-report from verbal interviews. And so we can see that our death tables and our self-report are really sort of can capture information that are in a lot of these OHDSI data domains, like conditions, procedures, measurements, etc. And so we label these algorithms to be high complexity. Similarly, we have OHDSI algorithms. So these all come from the OHDSI phenotype library, and they include, as you all are familiar with, logic leveraging condition domain, but also domains like measurements, procedures, etc. And so we deem these to be high complexity if they involve 3 or more data domains, and medium complexity if they involve 2 data domains. And then we also include phi code algorithms. These look at 2 or more occurrences of a condition code on distinct dates. And so we determine these to be medium complexity. And then we have our low complexity algorithms, which is just 2 or more occurrences of a condition code or any descendants. And so what we find if we run cohort diagnostics on these algorithms is that high complexity algorithms rely on a more diverse set of data domains to identify cases for cohort entry, but algorithms for the same disease often find similar condition concepts in the CHOP index events. So not surprisingly, then we find that high complexity EHR phenotyping algorithms result in increased GWAS power. And so we can see that by looking at the graph on the left, which looks at the number of cases by disease and algorithm. And we find that cohorts created with these high complexity algorithms, which are denoted by these red asterisks, have the highest number of cases. And then we also find on the right-hand side that these high complexity algorithms generally find a greater number of GWAS hits. And so hits are the number of SNPs that are significantly associated with the disease. And so this is really impactful to be able to identify sort of more potentially sort of disease-causing mechanisms along the genome, but we really want to make sure that these hits are biologically meaningful and we're not sort of just grabbing random, unuseful hits. And so one of the ways that we look at this is we look at the number of coding and functional GWAS hits. And so this graph here is showing the number of variants that are causal for disease and gene expression. And this analysis is known as GWAS EQTL colocalization. And so we can see we have two graphs here for asthma and type 2 diabetes. And we're asking how many variants are causal for both disease and for gene expression across a wide variety of tissues. And we find that high complexity algorithms generally result in the greatest number of colocalized variants. So these are the variants that are causal for both disease and gene expression. And separately, we also find that high complexity algorithms result in a higher number of novel hits on the coding genome. So these are hits in exons. And also this includes exons in sort of the most relevant genes for each disease. And so from this work, we can take away that high complexity algorithms generally result in improved GWAS outcomes, including increased power, hits with encoding and functional genomic regions, and colocalization with expression quantitative trait loci. And so we find that biobank scale GWAS can benefit from phenotyping algorithms that integrate multiple data domains, whether that be cause of death in the death table, self-report, measurements, procedures, observations, etc. And that curated repositories of complex, high-quality phenotyping algorithms are essential to advance the understanding of disease etiology. And so with that, I want to give a shout out, especially to the OHDSI Phenotype Working Group and the OHDSI Phenotype Library. These definitions were immensely useful in my work, and also just to the OHDSI community in general for, working so hard to promote this data standard. So thank you so much, and I'm happy to take any questions.